Good morning. In this new video, we are going to discuss cellular respiration. And this concept represents the second part of Unit 3 after having studied energy and enzymes. Of course, the two parts of this Unit 3 are very related because the study of transfer of energy and the study of enzymes speeding up biochemical reactions will be used extensively in this part on cellular respiration. As you know, before, respiration is the breakdown of bonds in food molecules that are rich in energy, like the glucose molecule, for the sake of production of energy for all vital processes in the cell that need energy. And we call it cellular respiration because it occurs inside the cell. And it's worth noting, of course, that the process of cellular respiration is a purely biochemical process, and it is totally different from the process of breathing, which is a mechanical, muscular, and skeletal process to inhale air in and exhale air out. The first slide includes a picture of the mitochondrion. And as you know, the mitochondrion is the organelle where cellular respiration occurs when oxygen is available. I mean, when it is aerobic cellular respiration. Of course, we have studied the structure of the mitochondrion, and the mitochondrion is one of the organelles that is double membrane. So it has an outer smooth membrane and an inner membrane that is folded, that is highly folded. We call every one of these folds a crista, and the total of all these folds is called cristae. So the inner membrane is highly folded, and it is highly folded to increase the surface area. And that surface area is important. It's important for that surface area to be large because it is the part which is the inner membrane of the mitochondrion that carries the electron carriers that will transfer the electrons in a sort of a chain. And at the end of this chain, we will obtain ATP molecules by having stored energy that is released from the transfer of electrons, as we explained before. So the inner membrane is important because it is the site of the part of the biochemical reactions that occur in aerobic cellular respiration. The matrix of the mitochondrion, which is the jelly-like substance inside the mitochondrion, is also important because in this matrix of the mitochondrion, we have important chemical reactions that will be described as we go over the process. So cellular respiration is a biochemical process in a living cell by which the cells break down the bonds in the food molecules, and they are covalent bonds, in order to be able to extract the stored energy. And then when this energy is extracted from the bonds, it will be stored in ATP molecules. Of course, the purpose of storing energy in ATP molecules is to prevent leakage of this energy to the outside environment in order to make the process more efficient. The goal of cellular respiration is to get energy for the vital processes. So when you get energy, you have to keep it. You have to prevent it from leakage because, as you know from our previous study on energy and enzymes, that if this energy is leaked, it will be leaked in the form of heat. And when it is leaked in the form of heat, most of this heat cannot be utilized, and therefore it will be dissipated to the environment, and dissipation of heat to the environment will increase disorder or will increase entropy. And therefore, ATP is considered an energy storage molecule in the cell. So it is an energy storage molecule in the cell, but at the same time, ATP molecule is the energy currency of the cell, meaning that at any time you can hydrolyze it easily to obtain energy. And we discussed the equation of hydrolysis of ATP before. If you have adenosine triphosphate, which consists of adenosine and 3 phosphate groups that are attached to it. If you add water to it, meaning that if you hydrolyze it, you will obtain adenosine diphosphate only. So from triphosphate, you get diphosphate. So from 3 phosphates, only 2 remain because 1 phosphate group is released because of the breakage of the bond that was between this phosphate and the 2 other phosphates. And when this bond has been broken, energy is released. So whenever we need energy, we hydrolyze ATP. Now, energy will be temporarily stored in the molecules of ATP, and we say temporarily because, as we have just explained, whenever energy is needed, ATP will be hydrolyzed and will be broken down to release energy. Of course, the ADP molecule has less energy than the ATP molecule because the energy in ATP is the energy in ADP plus this released energy. And because ADP has less energy than ATP, so we say that ADP is more stable than ATP. So ATP is more useful to the cell because it contains more energy. But concerning biochemical stability, ADP having less energy is more stable than ATP. This stored energy inside ATP is instantly available for the active cellular processes, and active means requiring energy. And is it important to say instantly available for active cellular processes? Yes, because this emphasizes the idea that ATP is the energy currency of the cell, meaning that it's very easily to obtain energy from ATP. And that's why ATP is an excellent candidate molecule for delivering energy to the cell because hydrolysis of ATP is easy. The energy released from ATP is easily utilized by the cell, so it is instantly available. And here in letter D, you have some examples of the active processes that might utilize energy in the cell, like, for example, the generation and the transfer of an electric impulse, the contraction of a muscle, whether this muscle is a skeletal muscle or a cardiac muscle or a smooth muscle, movement of cilia and flagella, as we have seen previously, because the motor proteins that lie at the bottom of the cilia and the flagella utilize energy from ATP to be able to move these structures. And the process of aerobic cellular respiration, in which you break down glucose in the presence of oxygen to release energy, is considered a highly exergonic process, meaning that it releases a huge amount of energy. And part of that energy, as we know, is utilized as an activation energy for the exergonic process, and part of it is stored in ATP. So the big amount of energy that is released from the breakdown of one molecule of glucose is stored in ATP, and 1 molecule of glucose is sufficient to give you from 30 is sufficient to give you from 36, there is something wrong, from 36 to 38 ATPs. So this is the amount of energy you can get from 1 molecule of glucose after aerobic cellular respiration has occurred. Now, this is what we call the ATP cycle. So here you have the nucleotide that consists of adenine. This is adenine, which is a nitrogenous base. And then here we have the 2 phosphate radicals that are related to, sorry, I apologize. So here we have adenine, the nitrogenous base that consists of 2 organic cyclic rings. And then here we have the sugar, the pentose sugar. And here we have 2 phosphate radicals. As you know, adenine is a nitrogenous base and the ribose is a sugar, and both together are called adenosine. So adenosine is adenine plus the ribose sugar. And you know from your previous study of nucleic acids that when you bind a nitrogenous base like adenine by a covalent bond to a pentose sugar to 1 phosphate radical, you obtain what we call the nucleotide. So adenosine diphosphate is very similar to the nucleotides that are present in DNA, except for the fact that you have 2 phosphate radicals instead of 1 phosphate group. And then if you add a 3rd phosphate group, then you are synthesizing adenosine triphosphate. In order to do this, you need an input of energy. So the process of synthesis of ATP is an endergonic process because energy is being uptaken and stored in the molecule. So we obtain ATP and water. Now, ATP has the same basic structure as ADP, meaning that it has adenine, nitrogenous base, and then it has the pentose sugar, and then it has 3 phosphate radicals. But the covalent bond between the 2nd phosphate radical and the 3rd phosphate radical is drawn as a squiggle like this and not as a straight bond, and this squiggle indicates that squiggle describes the shape we have just drawn on the picture, and squiggle refers to a high energy bond. So be careful, because when you draw such a squiggle, you are drawing a high energy bond. The reverse is also true, meaning that if you add water to ATP, then you are hydrolyzing ATP. So what you're actually doing is that you are breaking that squiggle and energy is released. So the process of synthesis is an endergonic reaction where the energy of the reactant is less than the energy of the product, whereas the process of hydrolysis is an exergonic process. Exergonic process because it releases energy, and here the energy of the reactant, which would be ATP in the case of hydrolysis, will be higher than the energy of the products, which are ADP and inorganic phosphate. And let's read the two statements that are present above and below the diagram because they are very important. So where does the energy to synthesize ATP come from? We have said that this reaction is endergonic and that we need an energy input. That energy input comes from catabolic reactions that are exergonic, meaning that whenever you have a catabolic reaction that releases energy, this energy can be utilized to synthesize ATP. Or in other words, more appropriately saying, we would say that this energy from exergonic reaction will be stored in ATP. Meanwhile, ATP hydrolysis will provide the energy to drive endergonic processes. In hydrolysis, energy is released. So this energy will be utilized by the endergonic formation of ATP. So we can say that exergonic processes drive endergonic processes. Whenever you have an exergonic process that releases energy, you can take this energy to activate an endergonic process. So you are sometimes required on the test to explain statements. So you might be required to explain this statement: exergonic processes like hydrolysis of ATP drive endergonic processes. Drive endergonic ones or endergonic processes. And the best and easiest example is this example of the ATP cycle. And as we have said before, one glucose molecule, when completely broken down under aerobic conditions, meaning in the presence of oxygen molecules, will result in the release of energy that is enough to be stored in 38 ATP molecules maximum, actually 36 to 38 ATP molecules depending upon the circumstances. And the biochemical equation is the following: C6H12O6, which is glucose or fructose or galactose, meaning a hexose, but a monosaccharide and a hexose monosaccharide. And of course, glucose is the most abundantly utilized because glucose is the monosaccharide that is the most abundantly present in carbohydrate molecules in the presence of 6 oxygen molecules. So one glucose molecule in the presence of 6 oxygen molecules, and therefore under aerobic conditions will result in the formation of usable energy, which is ATP. That is instantly, we might even add instantly usable energy. In addition, of course, we have here 36 to 38 ATP molecules per one glucose molecule, in addition to 6 water molecules and 6 carbon dioxide molecules, which are considered as, we do not say waste products, but they are considered as with a Y, byproducts of cellular respiration. This doesn't mean that they are not useful to the cell, because we will see that this carbon dioxide that is released from aerobic cellular respiration can partly be exhaled outside the body. The surplus is exhaled, but a good deal of it is converted to carbonic acid, and carbonic acid can give rise to bicarbonate, or in particular sodium bicarbonate, and both are utilized in the cell mainly for stabilization of pH, for homeostasis, or for what we call buffering of the cell. And when we say buffering of the cell, we mean making the pH of the blood more or less stable. Similarly, the surplus water is exhaled as water vapor, but part of the water, the greatest amount of the water, is recycled in the body and is utilized in several processes like dissolving substances, for example, in plasma, in the extracellular fluid. It is important for hydrolysis, and as you see, it is important for dissolution of substances, for transport, and for chemical reactions. It is important for hydrolysis. It is important for stabilization of the body temperature, and so it is important for excretion. So if we discuss the functions of water, we will find that water is extremely important in the body. And as we have said several times before, there are two types of cellular respiration. So we have the aerobic cellular respiration when oxygen is available, and the anaerobic cellular respiration if oxygen is not present. Of course, human beings and higher organisms in general are aerobic organisms. So higher organisms in general are aerobic organisms, meaning that they produce energy and ATP only in the presence of oxygen. But there are certain aspects of certain biochemical reactions in the human body that occur under anaerobic conditions, and we will see this. So what are the phases of aerobic respiration? We have three stages or phases of aerobic cellular respiration. The first phase is called glycolysis. The second phase is called citric acid cycle or Krebs cycle, referring to Sir Hans Krebs that discovered or described the Krebs cycle. And then finally, we have the electron transport chain that is associated with this process of oxidative phosphorylation, or phosphorylation associated with oxidation that we mentioned before. And it is worth noting that glycolysis in such a way is the first phase of aerobic respiration, whereas the electron transport chain and oxidative phosphorylation is the last stage of aerobic respiration. So electron transport chain that is associated with oxidative phosphorylation is the last phase of aerobic cellular respiration, and it is the phase where most ATP molecules are produced, as we will be seeing later on. It is worth noting that glycolysis occurs in the cytosol of the cell, and that it does not require oxygen. It does not require oxygen. So although it is part of a whole process that requires oxygen, yet for this first stage of aerobic respiration, which is glycolysis, and which occurs in the cytosol, oxygen is not a requirement. And therefore, we can say that glycolysis is the anaerobic phase of aerobic cellular respiration. So it is the anaerobic phase of aerobic cellular respiration. On the other hand, electron transport chain and oxidative phosphorylation is purely aerobic because it will occur only in the presence of oxygen. Why does it occur only in the presence of oxygen? Only, meaning that if oxygen is not present, this electron transport chain will not occur. This is because oxygen is the last electron acceptor in this chain of electron transport molecules. Oxygen is the last electron acceptor. So if oxygen is not present, the final electrons or the electrons will not be accepted and the whole chain will stop. The whole biochemical pathway will stop. So these are the phases of aerobic cellular respiration. On the other hand, if we don't have oxygen or if we don't have enough oxygen, the cell will carry out anaerobic respiration. Does this occur in the human body? Yes. For example, in skeletal muscles, when you are exercising heavily and the heart is pumping blood to the maximum to the muscles, and the lungs are getting in oxygen into the blood to the maximum. So maximum blood carrying maximum oxygen is reaching the skeletal muscle. However, the oxygen supply to the muscle, though maximum, is less than the oxygen needs. So the muscle must find a way to still get energy in this process of what we call, this is not absolute oxygen deficiency, but this is relative oxygen deficiency. Why? Because we do have oxygen that is being pumped to the muscle, but and it is a huge amount of oxygen, yet it is less than the needs of the muscles. So it can occur in the muscles under heavy muscular exertion. In such a case, anaerobic cellular respiration will start by glycolysis first, and that's why we have just said that glycolysis is the anaerobic phase that is common between aerobic and anaerobic cellular respiration. But in aerobic cellular respiration, at the end of glycolysis, we obtain a compound called pyruvic acid that results from breaking down glucose, and that pyruvic acid will enter a chemical pathway called Krebs cycle, where it will be further oxidized to further release energy. In anaerobic respiration, glycolysis will occur as usual and will produce pyruvic acid as usual, but in the absence of oxygen, there will be no Krebs cycle, no electron transport chain, and therefore no oxidation of pyruvic acid. And instead of this, pyruvic acid will undergo reduction. Reduction into what? Into either ethyl alcohol, in which case the process will be called alcoholic fermentation, ethyl alcohol, or into lactic acid, in which case the process will be called acidic fermentation. And here at this step, it will be important to remind you of certain chemical terms that are important to remember. First of all, reduction is gaining electrons or gaining protons. Oxidation is the loss of electrons or the loss of protons, as you know. So what happens actually in cellular respiration? In cellular respiration, we consider that glucose is oxidized. Why? Because as you will be seeing in the biochemical pathways of cellular respiration, that glucose will be losing hydrogen ions and electrons. Actually, glucose, for example, if one glucose molecule loses two hydrogen atoms, these two hydrogen atoms will be transferred in the biochemical pathway as two hydrogen ions and two electrons. Because if you add two hydrogen ions to two electrons, this makes the two hydrogen atoms. So along the biochemical pathways of cellular respiration, you will find that we will be moving on protons and moving on electrons with them. So if the glucose skeleton or the glucose carbon skeleton loses two hydrogen atoms, this means that it has been oxidized. So glucose is being oxidized. But as we have said, if we are carrying out aerobic cellular respiration and oxygen is present at the end as the final electron acceptor, so it will accept the electrons and will accept the protons from glucose. So one oxygen atom will accept those two hydrogen atoms to form one molecule of water. And that's why it's true that oxygen is a final acceptor of electrons, but actually it's also a final acceptor of the associated protons. And the combination between all this will result in the formation of water. And as we have said before, when we were discussing the active processes in the body in Unit 2, we said that as electrons are transferred from one electron carrier to another, they are being accepted at gradually lower energy levels. So the electrons are moving from relatively higher energy levels in a previous electron carrier to relatively lower energy levels in the next carrier. And the difference of energy is released, and this energy is not dissipated to the environment, but most of it is stored in ATP. So free energy is released, and this free energy which is released will be or will empower the synthesis of ATP. So if free energy is released from electron transfer, so we can say that the transfer of electrons, this is an important statement, the transfer of electrons along the electron transport chain powers ATP formation. So why does it power ATP formation or synthesis? Because the transfer of electrons along ETC is an exergonic process. It releases energy and it empowers an endergonic process, which is ATP formation. When one atom, say A, loses an electron, and this electron is gained by another atom B, so in such a case, A has been oxidized, whereas B has been reduced because it gained an electron. So we always have coupling between oxidation and reduction, because if substance A loses an electron, meaning that it gets oxidized, and that electron is not received by another substance B, which gets reduced, then if this substance B is not present, then this electron will return to A. So we always have coupling between oxidation and reduction. So this is what we call redox reactions. So oxidation of one substance is always coupled with reduction of another substance. Of course, it's easy now for you to describe the structure of the mitochondrion because we have said it several times. The mitochondrion is enclosed by a double membrane, as we said before, and it matches with the endosymbiotic theory. The outer membrane is smooth, whereas the inner membrane is folded into cristae or projections. And we've said that the inner membrane of the mitochondrion divides the mitochondrion into two compartments. This is the inner mitochondrial membrane. This is the inner mitochondrial membrane. So we have two compartments. The inner one, the inner mitochondrial compartment is called the matrix, and the outer one is simply called the outer compartment. So when we say the matrix, we mean all what is inside the inner mitochondrial membrane. And when we say the outer compartment, we mean the compartment that lies between the outer mitochondrial membrane and the inner mitochondrial membrane. Now, we have mentioned that the electron transport chain and the process of oxidative phosphorylation that is always associated with the electron transport chain occur on the cristae membrane or on the inner membrane, whereas Krebs cycle or the citric acid cycle mostly occurs in the matrix of the mitochondrion or the jelly-like substance that is enclosed inside the inner mitochondrial membrane. And this is a picture of a mitochondrion under the electron microscope. You can see, of course, the outer membrane. You can see the folds or the cristae, the inner folds, and you can see all around the ribosomes, the cytoskeleton, and other structures of the cell. Again, this is a picture of the mitochondrion. What's new on this picture is what we have said before, that the mitochondria have their own ribosomes that are important to synthesize proteins as usual, but you have to specify that they are important to synthesize their own proteins or to synthesize mitochondrial proteins so that mitochondria can be self-dependent. We also have DNA, and DNA is also important to regulate synthesis of the mitochondrial protein and to regulate the mitochondrial reactions. The presence of their own DNA, which is very similar to prokaryotic DNA in the fact that it, or in the sense that it is small, that it is circular, that it is multiple, and the fact that they have their own ribosomes, in addition to the fact of the presence of a double membrane. These three facts are three pieces of evidence to support the endosymbiotic theory. And this is again the ATP structure, and we can see that we have 3 phosphate groups enclosing 2 high energy phosphate bonds, but it is worth mentioning that the outer one has more energy than the inner one. And this is adenosine diphosphate, so you have only 2 phosphate radicals with a high energy bond. If you add energy, you get ATP. if you add energy to adenosine diphosphate, you get ATP. If you convert ATP into ADP, then you release energy and phosphate, and the energy is utilized to do work. So as we said before, ATP stands for adenosine triphosphate, and an adenosine triphosphate consists of adenosine and 3 phosphates. So it's worth mentioning that ATP is an organic molecule because it contains carbon and hydrogen. Where are the carbon and hydrogen? They are present in adenine and in ribose. This is just a diagram, but if you see the biochemical structure, you will see that there are carbon atoms that are bonded by covalent bonds to hydrogen atoms. So it is an organic molecule, and therefore the phosphates that are present in the ATP molecule are organic phosphates. And this is important because we have mentioned this before. If you add organic phosphate to a substrate, then the enzyme utilized is a kinase enzyme. But if you add an inorganic phosphate and you don't utilize phosphate from ATP, then the enzyme is a phosphorylase enzyme. ATP is unstable, as we said before, because it has a higher amount of energy. The 3 phosphates in it are all negative and they repel each other. So here, if we look, each one of these phosphates, as we have mentioned before, carries a negative charge and therefore they repel each other. It's true that they are bonded together, but they have tendency to separate, especially that we have high energy. So actually, you have 2 reasons for the instability of ATP. First of all, the negatively charged phosphate. This is one reason for instability. And the second one is the higher energy that is stored in the bonds. On the other hand, ADP is a more stable molecule. For the same reasons, you will have, but the opposite, meaning that you will have less repulsion between the phosphate radicals because you will only have 2 phosphate radicals instead of 3, and because it has low energy. And this statement is very important. The change from a less stable molecule, which is ATP, to a more stable molecule, which is ADP, is always associated with the release of energy. And this energy, of course, is used for active processes or cellular activities that require energy, like active transport. And at the same time, we are not only by hydrolyzing ATP, you don't only get energy, but you also get a phosphate that you can be used to phosphorylate another molecule. So release of energy from ATP is also associated with the release of a phosphate group, and therefore it gives you the possibility of phosphorylating a substrate. Now we start the first common step between aerobic and anaerobic respiration, which is glycolysis. And actually, in glycolysis, you split the glucose molecule into 2 pyruvic acid molecules. Now, this is the chemical formula of glucose, C6H12O6, but it's not the structural formula. And here we have more or less a mixture between chemical and structural formula. If you want to write it in a purely chemical formula, we will say that we have 3 carbon atoms, and then we have 4 hydrogen atoms, and we have 3 oxygen atoms. So if you, and the product is 2 of the pyruvic acid molecules, meaning that if you split the 6 carbon atoms into 2, so you will have one molecule of pyruvic acid having 3 carbons and another molecule having 3 carbons. And if you split the oxygen in glucose into 2, you will have 3 oxygen and 3 oxygen. But if you split the hydrogen or divide the hydrogen atoms into 2 to get the 2 molecules of pyruvic acid, you are supposed to get H6 and H6, but you only have H4 and H4. So here you have 2 less hydrogen atoms, and here you have 2 less hydrogen atoms. So you have 4 hydrogen atoms. So these 4 hydrogen atoms have been lost from glucose, and therefore the overall process of conversion of glucose into pyruvate is oxidation, because we have lost 4 hydrogen atoms, 2 and 2. So it is actually 4 hydrogen atoms, because we are supposed to have 6 in every molecule, but we have only 4. These 4 hydrogen atoms should be prevented from returning to the original glucose molecule, so they will be carried by a molecule called nicotinamide adenine dinucleotide. And in such a case, we will need 2 of them, so that each one of the NAD molecules carries 2 of the hydrogen atoms, and it carries them in a very specific way. So here we have 2 hydrogens that are directly carried on the NAD, and 2 protons that walk next to the molecule. Now, and we will see the details later on, but NAD is thus considered a hydrogen carrier that carries hydrogen that has been removed from glucose. And meanwhile, it is considered a coenzyme for the enzyme that stimulates or accelerates that reaction. And it is a coenzyme, but not a cofactor, because it is an organic molecule. So NAD is the short name for nicotinamide adenine dinucleotide, and it's clear from the name that it contains nicotinamide, which is related to niacin and vitamin B group, and adenine, which is the nitrogenous base we have just seen, and dinucleotide because its structure is similar to a nucleotide containing adenine, containing a ribose sugar, but not a phosphate. So glycolysis is a process that occurs in the cytosol of the cell, as we have said before. So the site of glycolysis is the cytosol of the cell, and glycolysis is an anaerobic stage of both aerobic and anaerobic cellular respiration. So it is a common stage between aerobic and anaerobic cellular respiration, and it consists of 10 steps, and the overall reaction is the one we have just seen on the previous slide, which is breaking down one molecule of glucose, which is a 6-carbon molecule, into 2 molecules of pyruvic acid or pyruvate, each of them, as we have seen on the previous slide, containing 3 carbon atoms. Energy, of course, is released because when the 4 hydrogen atoms have been separated from the carbon skeleton of glucose, energy was released, and this energy was enough to form or to be stored into ATP molecules. So every molecule of glucose will give rise overall to 2 ATP molecules. But here in number 3, we tell you that 4 molecules of ATP are released per glucose molecule and not 2. Yes, this is very correct. Why? Because Here, what we have on this slide is the overall result of glycolysis, meaning that the 2 ATPs are the final output of energy that is released. But actually, this molecule of glucose gives 4 ATP, gives energy that is enough to produce 4 ATP molecules, but 2 out of them are utilized as the activation energy of the reaction. We have said before that exergonic reactions use their own energy that they release to activate themselves or to activate the subsequent binding of the subsequent molecules. So therefore, the 4 ATP is the total energy that is released from one glucose molecule, the total energy that is released per glucose molecule. But 2 ATPs is the net ATP molecule number obtained. So 4 molecules of ATP are released, but the energy of activation is 2. So after subtracting the 2 ATPs from the 4 ATPs, the glycolysis of 1 molecule of glucose would result in a net gain of 2 ATPs. Okay, so we take 2 out of the 4 to activate the reaction to produce 2 pyruvate molecules. So the 4 ATPs are the initial released amount of energy, and the final or net is the 2 ATPs. And this is called the direct energy yield of glycolysis. The direct energy yield of glycolysis. So what is the direct energy yield of glycolysis? It is the net ATP molecule number that we have obtained per glucose molecules, 2 ATP per glucose molecules. Why are we saying this? Because if we say that this is the direct energy yield of glycolysis, this means that we have an indirect energy yield of glycolysis. This means that we have an indirect energy yield of glycolysis. What is the indirect energy yield of glycolysis? If we go back to this, we will find that the products of glycolysis are 2 pyruvate molecules, 2 net ATP molecules that have been obtained by or that have been obtained directly. And directly here means in the cytosol without being forced to go to the mitochondrion. But these 2 NAD molecules that have been reduced, because we already had this hydrogen carrier 2-NAD in its oxidized state, and then afterwards it acquired hydrogen and became reduced NAD. This reduced NAD has now acquired 4 hydrogen atoms. 2 of them have been attached to the molecule as hydrogen atoms, and 2 go hand in hand with the molecule as protons, having next to them their electrons. And these will go to the mitochondrion in case oxygen is present. So in case oxygen is present, these 2 reduced NAD will go to the mitochondrion and will deliver their electrons to the electron transport molecules on the inner mitochondrial membrane. And at the end of the biochemical pathway of the electron transport chain, we will find that we will obtain ATP molecules because of, as we said before, the electron transfer is an exergonic process. And we also have just said that this exergonic process drives the endergonic process of formation of ATP. how many ATP will be formed from every reduced NAD. From every reduced NAD, we will get 3 ATP indirectly. And when we say indirectly, we mean through the electron transport chain. So if you have 2 reduced NAD molecules, then you will obtain 6 ATP molecules. If you add these 6 ATP molecules that are obtained indirectly, so indirectly you have 2 reduced NAD molecules that will give you 6 ATP. So the total yield of ATP from glycolysis will be the 2 ATP molecules that are obtained directly plus the 6 ATP molecules that are obtained indirectly. Glycolysis occurs in the cytosol, as we said, and releases ATP without using oxygen. But if we don't use oxygen, this means that we will not be allowed to enter the mitochondrion, and that the energy yield of one glucose molecule will only be the direct yield, which is the 2 ATP molecule. Glycolysis seems to be a simple one-step process, but what you have seen on the slide, that is a one-step process, is just the overall equation of glycolysis. But glycolysis is actually a biochemical pathway that consists of a series of chemical reactions, and actually there are 10 biochemical reactions, and the first one will trigger the second one, and so on. So it consists of 10 steps, and each step is catalyzed by a different enzyme, as we have said in our study of the biochemical pathway. This process of glycolysis releases 8 molecules, even if we obtain energy from reduced NAD and if we enter the mitochondrion. So it is 1/4 of the energy stored in glucose only. What does this mean? This means that most of the energy of glucose is locked in the pyruvate molecule, and therefore pyruvate, which is the end product of glycolysis, not of respiration as a whole, still has a lot of energy, and therefore it has to join another biochemical pathway inside the mitochondrion. In which part? In the matrix of the mitochondrion, so that it gets further oxidized and releases further energy. So if there is no glycolysis to yield pyruvate, then Krebs cycle is not empowered, and the following step and the last step, which is the electron transport chain, will not be working. So glycolysis is essential to give you its end product, which is pyruvate, that will join Krebs cycle, and then the products of Krebs cycle will be triggering the electron transport chain. We come to the end of this video, and then we will be continuing respiration in parts 2 and 3 of cellular respiration. Thank you.